Okay, so today we are going to talk about parasitic weeds. Uh, these are the plants which are attached to the crops, and for them to complete their life cycle, they need a host, which happen to be the crop. So the learning objectives are basically three. The first one is that we want you to be able to identify parasitic weeds and their symptoms on the host. The second objective is to be able to understand the biology of the parasitic weeds. And the last one is to be able to understand the general control strategies. What you are seeing here with red flowers is the Striga asiatica, probably parasitizing on some grasses. Again, Striga asiatica in a maize field. This is actually in Gokwe. Let me admit somebody. This is okay. So this is Striga asiatica in the low veld. You can see at the bottom of the maize stalk, maize stem, just above ground level, you have these plants. They represent Striga asiatica. Here we have got the maize plant grown in a polythene bag infested with the Striga asiatica. These are the plants with red flowers. It's actually Striga asiatica. And the symptoms on the plants, you have leaf drying. These are the symptoms of Striga asiatica. Again, Striga asiatica in Gokwe. You can see the short maize plants. This is the effect of the parasite. Again, here in Gokwe, where we have the farmer holding this Striga. Again, the Striga plant, but you can see the maize is not is doing well. We have got a range of Striga asiatica plants or Striga asiatica strains. We have got white strain, which is this one on our left, and the red one on our right. This one is Striga forbesii, which is found around the Kadoma area and some parts of Kwekwe. Probably it could be found in other areas as well, but it tends to prefer red clay soils. This one is the Striga gesneroides. In this country, it attacks tobacco, but in some countries they find it on cowpea. Again, Striga gesneroides at flowering. Again, the same weed. This is the uprooted Striga gesneroides, showing the attachments to the tobacco. What you are seeing is a tobacco field which was affected by Striga gesneroides. You can see the tobacco leaves are drying, and this can affect the quality of tobacco. This is Electra vogeri, a parasitic weed. Here it is attacking bambara nuts. It has those yellow flowers. Again, Electra vogeri in bambara nut. This is mostly found in communal areas. Electra vogeri in cowpeas. This is actually in polythene bags. Somebody was doing an evaluation of cowpea genotypes to find out the impact of this weed across cowpea genotypes. What you are seeing here is the Kescuta campestris. It looks like a spaghetti. These are the thin stems which are spread all over another plant. Again, Kescuta campestris, which looks like a spaghetti. This one is Ramphicapa fistulosa, which attacks rice. Normally we find it in wet areas where rice is grown, especially traditional rice. That's where we find this weed. Well, what you are seeing here, it's a shoot parasite, the mistletoe, which can attack trees, but so far it's not a problem in this country, but in wild trees you may find it, and probably it plays a role in nutrient cycling. So this is the shoot parasite. If you want to know more about it, the mistletoe, a shoot parasite, you may read this paper written by Zimbabwean scientists. The bromborep or bangi species, that parasite with the bluish flowers, is found in some parts of Ethiopia, Saudi Arabia, and Mediterranean region. So parasitic weeds obtain their carbon and mineral salts from the host via ostoria, which is a certain structure which allows the passage of carbon and mineral salts from the host going to the parasite. The local name for parasitic weeds is witch weeds. This is actually the common name. Local names, we have Bise, Ruombi, and Isona, depending on where you are in Zimbabwe. Now let us look at the classification of plant parasites. We have what we call shoot parasites. Shoot parasites are attached to the shoot or to the shoots of the host, like the mistletoe, which attacks trees, or the Cascuta campestris, which looks like spaghetti. It also attacks the shoot of its host. On the other hand, we have got root parasites. These are attached to the roots of the hosts. For example, Striga asiatica, which attacks maize, and the point of attachment is the root. Electra vogeli in bambara nuts, it also attaches itself on the roots. This same applies to Striga gesneroids, which attaches itself to the roots of the tobacco plant. We can also use chlorophyll to differentiate different types of parasites. We have got what we call hemiparasites. These contain chlorophyll and are able to photosynthesize. For example, Striga asiatica. Then there are holoparasites. These lack chlorophyll. For example, the orobanchi. The Cascuta is somewhere between holo and the hemiparasitism. It has a bit of chlorophyll, but it's not a lot. Now, in Africa, we have crop yield losses, which are associated with the parasitic weeds. It was estimated that about 73 million hectares devoted to cereal production in Africa were being affected by parasitic weeds. This was a report by Mbub in 1994. Sobon in 1991 estimated a yield loss in sorghum which ranged from 6 to 21%. Whereas in maize, the loss went up to 40% as reported by Kim in 1991. Cereals and grain legumes, normally which are affected by parasitic weeds, do not produce grain. In table 1, we have got common parasitic weeds in Zimbabwe. The first one is Striga asiatica. This one can parasitize maize, sorghum, millet, and sugarcane. Another one is Striga forbesii, which I said can be found around the Kadoma area, some parts of Kwekwe. Probably it could be found in other areas, but on heavy soils, where it can attack maize, sorghum, millet, and sugarcane. Then Striga gesneroides attacks tobacco, but in some countries it has been observed to attack cowpeas. We have not yet got some reports in Zimbabwe where it is attacking cowpeas. Then we have got Electra vogeli, which can attack bambara nuts and cowpeas. Cascuta campestris normally attacks lucerne, although it has been found on some vegetable crops, tomatoes, I think, in Marondera. The other parasitic weed which I have not displayed there is the Ranthicarpa fistulosa, which attacks rice. So we have got more or less two plant families which contain parasitic weeds. The first plant family, known as the Orubangasi, has a lot of parasitic weeds. It has Striga asiatica, Striga forbesii, Striga gesneroides, Electra vogeli, Ranthicarpa fistulosa. Then we have got another plant family, the Cascutaceae, or sometimes referred to as the Convolvulaceae. This is where we find the Cascuta campestris. The Cascuta is the shoot parasite, which is a current problem when you are growing lucerne in some parts of this country, Zimbabwe. Table 2, we are showing the seed size. Here we just want you to appreciate how small the seeds are from the parasitic weeds. For Electra vogeli, seed length is 0.2 millimeters and seed weight is 0.3 micrograms. Striga asiatica, 0.33 millimeters length, and the weight is 3.7 micrograms. Probably the parasite with large seeds is the Striga forbesii, with 0.5 millimeters seed length, and the weight of each seed is 12.4 micrograms. But nevertheless, the seed appears like powder, dust, If you want to see the actual seed, you have to use a microscope. So that's how small the seeds are. Seed production, Striga asiatica can produce about 25,000 seeds per plant, whereas Striga foxii can produce 58,000 seeds per plant. And these seeds can last for up to 20 years being alive in the soil, which means that the parasitic weeds can give, serious problems. It may not be easy to get rid of these plants with the high seed production and that kind of seed longevity, which is up to 20 years. This is what you see under a microscope for Striga asiatica seed. This is for Cascuta campestris seed. This is what you can see under a microscope. So it may be easy to distinguish which is which, whether you've got Cascuta campestris versus Striga asiatica. Here I've got a map with those black dots. It shows the areas globally which is affected by Striga species. Here we've got a small dot in the US which is affected by Striga. Probably down here we have some dots, but you can see that this is an African problem, these parasitic weeds. Australia we have bits and pieces, in Asia, India, and so on. In Zimbabwe, we find these species almost across 5 agroecological regions, but they tend to be highly frequent in natural region 4, 5, and 3. Although in natural region 2 we find these parasitic weeds, but they are a serious threat, especially in low rainfall areas. Now let us look at the life cycle of the Striga. We have to start with the seed. Now when the seeds have been produced, they need to germinate. And the germination requirements is that first you need what is known as the preconditioning requirement. Preconditioning requirement is actually the exposure of the Striga seeds to moisture or to moist conditions, probably for a period of about 1 to 3 weeks, probably depending on the strains. And during this preconditioning period, the temperatures which are required are approximately 25 to 35 degrees Celsius. So if the seeds are exposed to these conditions for 1 up to 3 weeks, then they will be able to germinate. But before that, the host has to produce what are known as the germination stimulants. These germination stimulants are a variety of compounds. Some of them which have been identified are as follows. We have got what is known as strigol. Another one is sogolion. We have got sogolactone. We have got electrol. In fact, we have got quite a number of these compounds depending on the plants. In fact, most plants produce these compounds. So once you have these compounds and you have your preconditioned striker seed, then the germination process will begin. But then after the germination process, it should be followed by what is known as the attachment process. So let me stop here a bit and say that for this process, we need what is known as the pre-attachment process. Pre-attachment process has got two things. You need preconditioning, you need moisture, you need the temperatures to range from 25 maybe up to 35 degrees, and you need to expose the seed for 1 up to 3 weeks. The second pre-attachment condition is that you need the germination stimulant, which is produced by the host. Then we move to attachment and the post-attachment. The attachment process itself will require another product known as 2,6-DMBQ so that the attachment can take place. But it has now been realized that there are several what are known as hostoria initiation factors or substances that allow attachment. You call them hostoria initiation factors. And the 2,6-DMBQ shown there is one of the examples. There could be other compounds which are yet to be identified, which allows the process of attachment. So there's going to be a physical connection between the host and the parasite. And that physical connection is a structure which we call the hostoria. That will enable the host, the parasite rather, to get carbohydrates, some nutrients, and water from the host. This is an extractive process which may cause the host to suffer because it is losing a lot to the parasite. Thereafter, the parasite may remain underground for about 1 month, 30 days, whilst benefiting from the host. Then it will emerge, and when it emerges, it is hardly recognizable because these are very tiny plants. But at flowering, which occurs probably after 1 month from emergence, we begin to identify these parasites because they will be displaying red flowers. Then after that, there is now seed formation, and the process will have been completed. So this is what I was trying to refer to. You need a period of preconditioning. I have gone through this. I have actually talked about these things. You need the temperature, 25 to 35 degrees Celsius. You need the moisture. Of course, this period can vary 1 to 2 weeks or 1 to 3 weeks. Then you need the chemical signals in the root exudates. So this is the germination of Striga asiatica under a microscope. You can see these under a microscope. These are the seeds and the white radicles which are searching for the host. These are the radicles which get attached to the roots of the host. And here we now have attachment. This is actually the maize root, and you have your striga with its radicle now attached to the maize root, and at the point of attachment, this is what we call the ostoria. So these are the germination stimulants. In fact, there are quite several. We call them strigolactones. One example is strigol, which was isolated from cotton, although cotton doesn't really support the parasite. Sogorion and the sogolactone, I think these are from sorghums. And scientists have actually produced artificially strigol analogs. We've got the GR24. Probably the idea was to try and form or process a herbicide which could be sprayed to these seeds so that they can be fooled and germinate in the absence of the host, causing them to die. We also have electrol and ethylene. And as I have mentioned, these are just some of them. We also now need the contact with the host root. That stage is important, where the parasite now forms the ostoria. I have actually mentioned it as a contact point with the host. The ostoria formation requires 2,6-DMBQ or 2,6-dimethoxy-P-benzoquinone. Striga triggers the formation of this substance at the root surface, where other substances which may be of importance, which may play a role. We have got the phenolics, flavonoids, cytokinins, and the thidiazuron. Here we've got a picture showing the ostoria, or the connection between the host and the parasite. So ostoria is like a bridge between the host and the parasite. So it is used to extract water, carbohydrates, and mineral salts from the host. Now the period underground, the parasite is small and lacks chlorophyll. It is fully dependent on the host. In other words, that is the time when it can cause serious damage. It can also, you cannot control it using mechanical methods because you cannot see it. That's why it is difficult to control. 30 days after germination, the parasite is underground. Then from 30 days to 40 days after emergence, it flowers. Then it forms seeds, and the life cycle is completed. Here we are going to focus on the effects on the host. Number one, the effects can be variable depending on the host genotype. Sometimes the genotype can express the effects of these parasites, sometimes it doesn't. If it doesn't, we say the host is tolerant. So we can have undetectable effects, or the host may actually die. So some cereals are resistant or tolerant, whereas others are susceptible. Usually the infected cereals show low stem weights and low grain yield. This is the effects of striga on the maize. You can see leaves drying and lodging of maize. Here you can see the infected maize showing wilting symptoms, even under wet conditions. These are typical symptoms of striga infection. Here we've got SC513 maize cultivar, which was affected by striga on heavy soils. Again, you can see some striga plants near the maize. Now, striga can cause standing of internode elongation. Wilting symptoms, even under wet conditions, as you have seen on the pictures. Probably you should know that there are similarities between the physiological effects caused by drought and those caused by striga. Striga tolerance may mean that the cereal could be producing a lot of abscisic acid, which causes the stomata to close. Striga reduces the rate of photosynthesis in the host. This is how it brings about its reduced growth in the cereals. But Striga tolerant cultivars may be capable of maintaining photosynthetic rates. Striga predisposes sensitive crop cultivars to photoinhibition or light impairment of photosystem 2. Striga infected cereals show lower stomatal conductance than uninfected plants. So what it means is that if the striga attaches itself to the host, that host will tend to close its stomata, so that because it has something to do with abscisic acid and the control of which controls the opening and the closure of the stomata. So once the stomata is closed, maybe carbon dioxide limitation will reduce growth. Please note that we can measure photosynthesis using those instruments: portable modulated chlorophyll fluorometer, infrared gas analyzer, and porometer. You may need these instruments when you are studying the effects of parasite on the host. It is possible that striga could be producing phytotoxic effects on the host. Now, as far as Alectravo jelly is concerned, also Cascuta campestris, the host is affected because of redirection of the carbohydrates. Most of the carbohydrates are directed to the parasite, and this reduces the growth of the host. Because these Alectravo jelly and Cascuta campestris form what we call very strong sinks for carbon and nitrogen. But for striga cereal interaction, the parasite can make significant contribution to its carbon requirements because it can photosynthesize. And if you look at it, the parasite biomass is very small, and movement of carbohydrates may not account for the impact of striga on the cereals. In fact, there's also evidence that only 20% of carbohydrates are directed to the parasite. So with the striga, probably it is affecting the host through photosynthetic through the photosynthesis, which is reduced in the host. So you may have low leaf area and low rates of photosynthesis, which could account to other carbon losses. Low stomatal conductance, this limit, carbon dioxide entry, and probably increase of photorespiration which tends to destroy carbohydrates in cereals. This is the root which Striga asiatica or Striga species could be using to damage the cereals. There are also changes in plant growth regulators. Once there are changes, this can affect host biomass allocation and architecture. For example, infected plants have higher root shoot ratios and shorter internodes relative to infected controls. You can also have changes in abscisic acid, gibberellins, cytokinins, which can cause architectural changes in the plant. ABA can also cause low stomatal conductance. So these disturbances, physiological disturbances, which occur in a host, could be induced by the striker. That's why the impact is so high in cereals, being caused by very small plants. So this is it on the introduction to parasitic weeds. What we now want to do is to look at the control of parasitic weeds.